Laboratory session this afternoon, we're going to discuss the following or do the following exercises. I don't want to take your time, so I pre-recorded this and just please follow the instructions, okay? So for the first exercise, that's exercise number 9. Here we're going to simulate the conventional PCR by detecting a disease that you call brucellosis. In bacteriology, you've learned that brucellosis is a bacterial infection caused by your brucella that spreads from cows, sheep, goats to people, and people are infected by eating raw or unpasteurized dairy products. Okay, so these are the materials that you need. That's there: the isolated DNA, the primers, PCR buffers, the NTPs, water, Promegatac, PCR tubes, agarose powder, TAB buffer, loading buffer, ladder, pipetteur tips, and then of course your equipment. And the specimen that we're going to use are your fresh milk. And the procedure that we're going to do here is of course DNA isolation. Next is DNA amplification by PCR, and detection of amplicons by gel electrophoresis. Okay, those are the 3 procedures we're going to do. So, DNA application by PCR. First, you have to prepare the PCR master mix. So, this master mix consists of your nucleotide bases, which is the sources of bases for extension, the enzyme DNA polymerase for the promoter extension, buffer, magnesium, which is the cofactor for the enzyme DNA polymerase. This PCR master mix does not contain the DNA template or the DNA source, and of course it does not contain primers. Okay, so this is the basic content of your master mix. So prepare this as the first step. Okay, the second, use the PCR amplification profile accordingly. So to detect the Brucella group, you need to denature it by 95 degrees Celsius for 35 seconds, annealing 56 degrees Celsius 45 seconds, extension 72 degrees Celsius and 45 seconds. And the number of cycles are 30. To detect naman Brucella militensis, we need the following: the initial denaturation of 94 degrees Celsius at 2 minutes, and then a denaturation step of the 95 degrees Celsius for 20 seconds, annealing of 55.5 degrees Celsius, and 20, that's for 20 seconds, extension of 70 degrees for 30 seconds, and the number of cycles is 35. Okay, so that's your amplification profile. And then for gel separation, use 1.5 TA agarose gel stained with ethidium bromide. Use 10 microliter of the PCR product with 2 microliter of your loading buffer. Perform the gel electrophoresis at 100 to 120 volts for 30 minutes. The negative control should not have an amplicon. The positive control shall be the fragment sizes of 600 BP for the Brucella group of specific and B 731 BP for the B metlitenensis. In addition, B Swiss has expected results of 285 BP and B Ovis 976 BP. If possible, have the amplicon sequence. No. So please enter the virtual laboratory by clicking this link. I'll provide you with the link in this in the description of this video. Just follow it. Okay, so that's for a laboratory exercise, and you have to answer the exercise sheet, of course. Okay, for exercise number 12, we're gonna do PCR naman, RT-PCR naman, no? So we're gonna detect the Newcastle disease, no? So what is Newcastle disease? Newcastle disease virus is a zoonotic virus that belongs to the family Paramyxoviridae, and it infects all species of birds. NDV have virus three types: the virulent lentogenic strains, the moderately pathogenic mesogenic strains, and the highly pathogenic velogenic strains. So lentogenic strains are the one used for NDV live attenuated poultry virus vaccines. So these are the known primers of these Newcastle disease, and detection of this can be done through RT-PCR. Okay, so sorry, let's proceed. There. So in this RT-PCR, we're just going to do a one-step RT-PCR. So the viral RNA that will be extracted from sample material will be converted into complementary DNA using reverse transcriptase and amplified thereafter by the TAP DNA polymerase. That's your RT-PCR. So these are the steps. Prepare the master mix. So these are the contents of our master mix. Perform the DNA synthesis reaction at 42 degrees Celsius for 14 minutes, and then add inactivation of RTAs at 95 degrees Celsius for 7 minutes. And after this, perform the PCR amplification using the profile listed, no? A denaturation step of 94 degrees Celsius, 30 seconds, annealing of 33 degrees Celsius for 30 seconds, extension of 72 degrees Celsius for 30 seconds, and a number of cycles of 55. Okay? Okay, so exercise number 12. Let's go to the part of gel separation of exercise number 12. Use 2% agarose gel stained with ethidium bromide and 10 microliter of the product. Use also 2 microliter loading buffer, 2 microliter of 100 BP DNA molecular weight marker. Gel electrophoresis should be performed at 100 to 120 volts for 30 minutes, and analysis should be done using an automated photo documentation system. Okay, so what we're going to do is you need to watch this video for you to be able to answer the exercise in number 12. So just please click this link. No, I'll send you a copy of that link as well. So then answer exercise number 12. Okay, so that's for the laboratory exercises. Okay, if you have questions, just send me or my partner an email, no, or send me a message. So thank you.